हॅलो. गुड मॉर्निंग कार्तिक. कार्तिक, गुड मॉर्निंग.
कांतीमती मॅडम, तुमचा आवाज खूप कमी येतोय.
हा,
तुमचा आवाज हळू येतोय.
आता येते व्यवस्थित?
हा, आता येतोय. हो.
हा, ओके. हा, तिथे कार्तिक मला ऐकू येत नाहीये, तर मला शेअर करायची स्क्रीन. हा, बर 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 मग मी शेअर करू का स्क्रीन? हा, ते मला ते कळत नाहीये, म्हणून मला कोणाची तरी मदत लागणार आहे. गुड मॉर्निंग डॉक्टर सिंग. गुड मॉर्निंग. हा, नाही नाही, मी सिंग आहे त्याच्यावरती, त्यांना गुड मॉर्निंग केलं. हो हो. हा, बोला. हा हा हा, गुड मॉर्निंग कार्तिक. Now I have to share the screen for that book release. How do I do it? Huh. Okay, yeah, sure. Okay, yeah. Thank you so much. Yeah. Good morning. Now tell me, I'm there. How do I share this screen? For you to see.
कांतीमती मॅडम, तुमच्या आवाजाला अजूनही क्लॅरिटी नाही आहे. थोडंसं तुम्ही जवळ घेऊन बसा किंवा अभी somebody sharing a screen, no? That's why you are not allowed to do that.
Okay.
Instead of others to be sharing.
Good morning, sir. Whether I am audible?
Am I
Kanti
audible,
Bati,
sir?
yes, you are. Madam, I'm giving you a call. Please pick up the phone.
Yes, sir. Huh. Sir.
Dr. Dhakefakar, you are on mute.
Yeah, I think Dr. Vidya Madam has also joined. Good morning, Madam.
Good morning. Yeah, I was probably the first one to.
Right, I know. Yes, right.
But I
We
just
are,
stopped the
right,
video and muted.
right. Yes. So Kakodkar sir will also be joining at 9:55. He has said that.
Yeah.
I just confirmed. And Karmalkar sir will be joining little late.
Okay.
He has some other function, so he will finish that and join here.
Join later. That's okay.
Yeah.
I think he has to that way speak little later only.
Right. He has to.
Yeah, I think.
Hello, this is Kakodkar here. Can you hear me?
Yes, sir, we can hear you loud and clear. Thank you so much. Very good morning to you, sir.
Thank you. Morning. And my picture is there?
Yes, sir. Absolutely
Okay.
clear. Right, sir.
Good, good,
Yes,
good.
sir. Right.
So, compliments to you,
Sir,
Founders Day.
thank you so much, sir. Compliments to all of us.
Yeah.
Right, sir. Right, sir. So we will be starting in couple of minutes, around
Yeah.
10 o'clock. Right, sir.
Take your time. Take your time.
Yes, sir. We will be.
Sir, we are live. We are ready to go. Kantimati madam, you can start.
Yes. A very good morning to everyone. On behalf of the director, NSCS ARI, I extend a hearty welcome to one and all to a very pleasant morning of many firsts and dual celebrations. The COVID-19 pandemic has brought about several changes in our working style and behavior. The Agarkar Day function or the Founders Day function is one such. In tandem with the Government of India guidelines of COVID appropriate behavior, we are all together, yet severally, on a virtual platform to celebrate the Founders Day function in a very novel way. This year is truly special for all of us. At ARI, as we have just entered the 75th year of the NACS Research Institute. That is the Platinum Jubilee year. The Department of Science and Technology, under whose wings NACS ARI is nestled, is celebrating its Golden Jubilee, giving us an additional purpose to extol our achievements. On that note, I now request Dr. P.K. Bhattacharya to present the Director's report.
Good morning. It gives me immense pleasure to be here to present the annual report for the year 2019-20. Respected Padma Vibhushan Dr. Kakodkar sir, who is President of MSCS, Honorable Vice Chancellor of SPPU, Professor Ditin Karmalkar sir, the Vice President of MSCS, respected Vidya Gupta Madam, Secretary of MSCS, respected Dr. Ghaskar P, the Treasurer of MSCS, my fellow colleagues, past and present scientists, students, and the members of the Agarkar Research Institute's family. It gives me immense pleasure to be here in front of you today on such unique occasion. And as Dr. Kantimati said, this is unique because Agarkar Research Institute has just entered into 75th years of existence. We are under the umbrella of Department of Science and Technology, who is celebrating the Golden Jubilee year this year. We observed 60th death anniversary of Dr. S.P. Agarkar on 2nd of September 2020. This is the first time we will be celebrating the Agarkar the Founders Day ceremony online, and there are several such challenges that we faced for the first time in the view of the COVID-19 pandemic. So there are lots of firsts this year, and continuing with this, I believe that this is the nice opportunity to take a look back on what we have done, look back at the past year with satisfaction that we were up to the challenge posed by the COVID-19 disease, and we did well. We don't want to be satisfied. We don't want to be contented. We want to be ready to face the next step, take next step to face newer challenges. So with that view, I would like to present before you the salient features of whatever research achievements that we achieved over past 1 year. And when I am saying that, I just want to let you know that within a short period, it would be very difficult to include all the highlights. So I would be touching at one or two random highlights from each of the thematic areas of the research carried out at Agarkar Research Institute. The mission of the research at Agarkar Research Institute is to conduct basic and applied research in life sciences and harness the genetic diversity of microbes, plants, and animals towards cleaner environment, sustainable agriculture, and better health for the masses. So the research at ARI deals with plants, it deals with microorganisms, it deals with animals, and also it deals with the nanomaterials. However, this research is mainly concentrated on 6 thematic areas. These thematic areas include biodiversity and paleobiology, bioenergy, bioprospecting, developmental biology, genetics and plant breeding, and nanobiosciences. We investigate biodiversity with several aims. It is just not to include the taxonomic novelty, but our major aim is to prevent the biopiracy. India is blessed with tremendous biodiversity. We have biodiversity hotspot in the Western Ghat, we have biodiversity hotspot in the Himalayan region, and also northeastern India, some part of it. So when we have such natural resources, developing nations or developed nations have exploited these natural resources, and to the tune that we can say that there has been tremendous biopiracy. Now we want to protect our natural resources, and to do that we must know what we have. We can protect only what we know we have and not what we don't know. Keeping that in mind, the scientists at Agharkar Research Institute have undertaken a tremendous effort to document the diversity associated with such hotspots. Now, when we are doing that, we want to document all kinds of taxa, all kinds of species diversity that is there. And in addition to that, we want to also document some of the novel taxa or novel species. We also want to conserve the endangered species. And in addition to that, whatever are the natural resources, we want to explore them for industrial applications. So keeping that in mind, we have been performing a research. Out the assessment of the local endemism of the flowering plants in the northern and western Ghats and Konkan region of India. So in this particular one, our scientists have carried out or compiled the checklist of the local endemic flowering plants of the northern regions of the Western Ghats. And while doing that, they have documented more than 180 taxa and also therophytes as the successful living strategy. Now, when I'm talking about that, what exactly our scientists have been doing is A, document the species diversity, and B, report any novel taxa whenever they are present there. And this has been a significant contribution to the taxonomy of the subject. And in addition to that, it is also building a database for the nation. Now, whenever our scientists have been performing these studies, their studies have become very important for one very important reason. Earlier, the plant biodiversity studies were mainly based on the phenotypic characteristics. They were mainly based on the morphological characteristics. But now our scientists have started making a lot of efforts to use the advanced molecular tools. They have been looking at some of the important biomarkers. They have been sequencing the whole genome. And based on that, they have been identifying various plant species which have been earlier unknown to us. In addition to that, our scientists have been successfully doing a lot of research on diatoms also. And this is a very important area where Agharkar Research Institute has established as one of the unique center, as not many people have been doing that. These diatoms have enormous applications as environmental pollution indicators. They have applications in forensics. They also have applications in bioenergy. And as a result, these diatoms have become a very important tool. Our scientists have developed a large collection of diatoms, and they have been documenting a lot of diversity of diatoms. In addition to that, our scientists have been reporting a lot of species of the plants which were earlier unknown. And as a result, ARI has established as a center of excellence as far as the plant diversity is concerned. In addition to that, our scientists have also been working on a very unique system, and that is lichens. The reason I call this as a unique system is because there is a algal component and there is a fungal component which come together, establish a relationship in which they form a lichenized form, and this lichenized form can result in production of some of the biomolecules, some of the metabolites, which can be of enormous medicinal importance. Lichenization in nature takes a lot of years to grow, however, a lot of years to take place also. However, our scientists have developed a technology to culture these lichens in our institute at much faster rates within few weeks. Now, if at all you find any lichen culture which is producing medicinally important biomolecules, then obviously we don't have to wait for many years to get the significant quantities of that, but within few weeks, our scientists can culture these lichens and they can they can produce the biomolecules of our interest. Now, while doing that, what is important part is to have a large collection of lichens, and our scientists have been investigating them, and they have also started using molecular tools to investigate this. And as a result, our scientists have been able to find out or identify important uh, important species lineages in Indian Flavopuctalia and Puctalia species of lichens. And this is really a very important contribution as far as the species diversity of Indian Parmeliaceae family of lichens is concerned. In addition to that, our scientists have also been carrying out lot of fungal diversity studies with respect to fungi. Now, there are two types of fungi that our scientists have been working on. One is aerobic group of fungi and another one is anaerobic group of fungi. And this is a very interesting piece of work that our scientists are doing. And the reason I say that is because the aerobic fungi, for aerobic fungi, we have the National Facility for Fungal Culture collection, and this fungal culture collection houses a largest collection of fungi, and majority of them are aerobic fungi. The scientists who are working in this fungal culture collection are also investigating diversity of fungi. They are documenting a lot of novel species of fungi, and one such genera, newly discovered genera, has been reported and named as Srinivasanomyces kangraensis. Now, this is the fungus which was isolated from Kangra Valley, and this fungus has been named after tremendous contribution made by Dr. M.C. Srinivasan, who also happens to be one of the life members of MSCS. There is another interesting aspect of the fungal diversity, and that is related to anaerobic fungi. In anaerobic fungi, our scientists have been doing a lot of interesting work. These anaerobic fungi are of importance because they have the ability to degrade lignocellulosic biomass. These fungi are mainly present in the rumen of the ruminating organisms, animals. Now, these fungi have been very difficult to work with. However, our scientists have now become expert. They have developed certain techniques and expertise by which they can isolate, identify, characterize these fungi, and they have been also documenting new genera, new species, and such the pictures of two such novel genera are presented in this particular slide. Now, why this contribution is important? Because of the difficulty of cultivation of anaerobic fungi, their taxonomy has been lagging behind. However, our scientists have been doing a wonderful job in isolating, identifying, and characterizing these fungi, use advanced molecular techniques to investigate these fungi and explore these fungi for various applications, especially in the field of bioenergy. And that is why these fungal cultures are of very much importance to our research, and our scientists have done wonderful job. Now, when we are investigating such biodiversity, it is not only for documenting novel taxa. Sometimes it is possible that some species have become endangered. Such species then needs to be cultivated in the laboratory, and then they need to be again grown in the nature. Now, for that purpose, it is very important to carry out ex situ cultivation. Now, when we are using the tissue culture technique for cultivation of such endangered species, our scientists have also found out that this technique can be used for the cultivation of orchids. Now we all get fascinated by wonderful pictures, wonderful colors of orchid, how beautiful they look. And our scientists have now started and they have established a technique for epiphytic orchids. They can be cultivated in our laboratory at much faster rate, much more efficiently. And then these can be then developed in the form of a nursery. Very soon, Agartha Research Institute will be having a nursery of such orchids which have been cultivated ex situ in our institute. Now, when we have talked about fungi plants, also our scientists have been investigating the diversity studies on yeast. And these yeast are very wonderful organism. Our scientists have identified thermotolerant or thermophilic yeast as one of the important group to study. And one of the reasons for that, these yeast can be used for the fermentation of ethanol. Ethanol can be used as a biofuel, and our country needs biofuel to satisfy our ever growing need for the energy. And the yeast can be considered as a renewable source because they can grow very fast. But thermophilic yeast are special because their fermentation can take place at a higher temperature. When you are growing yeast at high temperature, the ethanol can evaporate, and such vapors of ethanol can be then collected and condensed, and ethanol can be segregated. When you are cultivating ethanol at in fermentation process, as the concentration of ethanol is going up, this becomes toxic to the yeast cells which are producing it, and as a result one can have lower yield. But thermophilic yeast will not face this challenge, as the ethanol is being continuously removed from the fermentation broth, and as a result the ethanol toxicity to the yeast could be very less. Our scientists have been doing a very good job, and this particular research is definitely in consistence with the national biofuel policy that set bioethanol blending target of 20%. And for that purpose, we definitely need newer technologies for production of ethanol. Continuing with this line, I would like to tell something about bioenergy. Now, in bioenergy, we have been doing some research for many years, and this research has been for the production of methane or biomethanation of rice straw. Now, rice straw is an abundant and cheap substrate from which the methane can be generated. Now, if at all it has to be generated, Agartha Research Institute has developed a technology which does not require any pretreatment using energy intensive heat process or environmentally polluting NaOH or alkali treatment. Our our process, that is the process developed by Agartha Research Institute, uses anaerobic fungi which can efficiently degrade rice straw and produce methane in very economical way. But our scientists have not stopped at this. Now they have developed a similar process for the production of hydrogen. And recently we have read in the newspaper that hydrogen fuel cell based car has been successfully tried by KPIT Engineering Limited. And KPIT now has collaborated with our institute, and for them we are developing the process for biological production of hydrogen. I cannot reveal many details at this particular time, but significant progress has been made, and very soon I believe that there would be a pilot scale trial conducted in our institute campus for the production of hydrogen in scale up studies. Another area which is very important is a bioprospecting. Whatever plant resources that our scientists have been investigating, these plant resources are also exploited for their ability to produce biomolecules or phytochemicals which can have medicinal application. And one such application is to cure the inflammation associated anemia. Now, for that, there has been a plant which is Tinospora cordifolia, or guduchi as it is colloquially known. And our scientists have found out that extract of these plants are efficient in controlling the inflammation associated anemia. Now, our scientists have published this work. They are in a process of identifying the active principle But this is not the only approach that they have been following. For other such applications, such as acetylcholine esterase enzyme, which is involved in the the enzyme hydrolyzes acetylcholine into acetate and choline at a cholinergic synapses, and this particular enzyme has to be inhibited. This is very important for the treatment of Alzheimer's disease. Now, if at all it has to be done, there has to be a certain compound which can efficiently do that. Thiosemicarbazones are such compounds which are of plant origin and which have been efficiently doing this job. Now, our scientists have started a newer process where they have started modifying these particular compounds, and because of modification they are able to get insight into structure activity relationship, that if at all the compound is there and if at all we make modification, whether we can identify what particular functional group is responsible for the activity and whether that activity can be successfully improved. And yes, our scientists have made such important modifications in such chemicals to improve the activity which inhibits the enzyme, which can be an important lead in the treatment of the Alzheimer's disease. Another such approach our scientists have been using is to synthesize these derivatives as well as chemicals which were originally identified from plant origin, so that the process can become more efficient and they can identify the application for such. And this is one such example where phenanthridine 1,4,6 trione epoxides have been used as antimicrobial agents against the bacteria such as Staphylococcus aureus. And they have identified that there are certain targets which can be attacked by this compound and which make these organisms susceptible. And after these bioinformatic insights, our scientists have then modified the compounds to improve the efficiency of docking and thereby improve the efficiency of antimicrobial activity of these compounds. Now, after bioprospecting, there is another area which is developmental biology. And in this particular area, our scientists have been doing wonderful job. Now, this is the area where our scientists have been doing mainly the fundamental research. However, that research has been having some important applications in future. There are three interesting models which are being investigated by our scientists, and they include Hydra, they include zebrafish, and they include Drosophila. As far as the Hydra is concerned, our institute has a unique place as not many labs have been working on this particular model. In this particular model, our scientists have been investigating various signaling pathways for regeneration in Hydra. Now, Hydra is a unique organism because of its ability to regenerate. Not many animals would have this kind of ability. As Hydra has this unique ability, it is of interest to find out which components of the signaling pathway play an important role in this regeneration. And as can be seen from these particular slides, our scientists have identified three new components, which are vascular endothelial growth factor, fibroblast growth factor, and these fibroblast growth factors, they are FGF1 and FGF1 receptor and VEGF receptor 2. Now, what is of interest to find out is our scientists have done some experiments, and in these experiments, they were able to show that if at all you inhibit the receptor 1 of FGF and VEGFR2, then this blocking of these sites would lead to the delayed head generation. Now, if you take a look at this particular control, you will see that even if the treatment is given for 48 hours, now in case of control, after 24 and 48 hours, the Hydra regeneration starts and you will see the head formation taking place. The solvent control also does not affect the head formation. But if at all you carry out the treatments for the inhibition of FGFR1 or VEGFR2, you will see that the head formation is getting delayed. And this is a very interesting observation made by our scientists. Now, these results indicate that VEGF and FGF, they play important role in the regeneration of hypostome and tentacles in Hydra. Now, what is the significance of this study? The signaling pathways which and the pattern forming mechanisms, they are conserved through the evolution. Now, similar molecules may be present in the more complex organism, and they may play an important role in tissue and organ regeneration. Now, if at all that is a possibility, then such insight can be used to study their homologs in the more complex organism and ultimately study whether the regeneration of at least some of the organelles is possible or not. Such is a very interesting piece of work that is being done by our scientists, and this has come from Dr. Ghaskarbi's lab. And I must say that Dr. Ghaskarbi has retired, but he is not tired, if I may use that cliche. And he has been very actively pursuing and publishing and leading by example. And I must say that the entire group of developmental biology has accepted the challenge in last year and half, and they have increased the number of publications that have been coming out of this group, and now that is definitely going to get a very good recognition for this group in a very near future. Another area is nanobiosciences, and in this is one of the bigger areas, bigger groups that has been working in Agarkar Research Institute, and their research has been mainly related to a lot of applications for industries. Now, it's very difficult to include all the highlights of the research being carried out in such big department, but some of the important highlights that have been shared by the scientists in nanobiosciences are enlisted here. And one such is antimicrobial nanomaterial that they have developed for disrupting the biofilm forming bacteria. Now, biofilm has a very important role in environment as well as in health. And the reason I say that is because biofilm can make any kind of treatment very difficult. If an organism in suspended form is susceptible to antibiotics, in biofilm or sessile form, it can definitely be resistant to such antibiotics. And then treatment of such diseases, especially cystic fibrosis like diseases, could be very difficult. Our scientists have developed certain nanomaterials which are based on silver and copper nanoparticles, and these nanoparticles are very effective in disrupting the biofilm of Pseudomonas. As you can see in this particular picture of confocal microscopy, you will see that the nanoparticle formulation at a very low concentration, around 12 ppm, is able to disrupt the biofilm. And this is possible not only by the physical damage that the biofilm can cause, but it can also disrupt and manipulate some of the gene expressions in the organisms also. And the scientists have done wonderful studies in which they have shown the upregulation of some of the altered metabolism and the down regulations of many of the genes, which also include the quorum sensing genes. And these are the genes which play an important role in biofilm formation. And as a result, you can see that the biofilm disruption is possible because by the use of such nanoparticles. Nanoparticles have also caused physical damage to the biofilm community in addition to gene expression. And you will see that their applications can be in the medical as well as in the environmental setting, and it can be in for nutrient recycling also, and it can be for xenobiotic degradation also. Another nanomaterial which has been used in agricultural application has been zinc nanoparticles, which actually increases the fertilizer use efficiency by 8 folds, and that is very, very significant. And it also improves the grain zinc content significantly without compromising the yield. And zinc as a micronutrient is very important, and you can see that use of such nanomaterial is going to be extremely important in improving the nutritional properties as well. In addition to that, you will also see that the nanomaterial have important role to play in controlling the diseases caused by different bacteria in vegetables, in fruits. And one such example is of copper nanoparticles that can control the bacterial blight disease in pomegranate. Thalia is the commonly known or vernacular name of the disease. And you will see that the nanoparticles of copper, they are able to inhibit these organisms. You can see the practical you can see the picture where you can see the stomata on the leaf surface showing the dead bacteria, and you will also see how effectively the organism is inhibited by use of these nanoparticles. You can also see that some of the important findings is there are different nanoparticles. You will see the copper nanoparticles, zinc oxide, magnesium oxide, copper oxide, etc. And you will see that copper nanoparticles have been the most efficient as far as the antimicrobial activity is concerned. And the sixth group is genetics and plant breeding. And this group also has been doing wonderful job as far as the development of the varieties of wheat, development of varieties of soybean are concerned. And one such, the most important part of this particular one, as far as the uses for farmers is concerned, is as follows. The varieties developed by the scientists at Agharkar Research Institute are mainly disease-resistant varieties. They are high-yielding varieties. They reduce the reliance on the fertilizer usage. They reduce the reliance on the pesticide usage, and they also require less of the water. That is, irrigation requirement is significantly lower as far as the varieties developed by Agharkar Research Institute are concerned. Now, this is not the only interesting features of the varieties developed by Agharkar Research Institute are concerned. We have the wheat variety, which is MSCS 4028. It is a biofortified variety. And the reason we say it is a biofortified variety is because it is rich in the protein content, it is rich in the iron content, and it is rich in the zinc content. Now, this biofortified variety can have an important application in fighting against the malnutrition. And this is also in consistence with the policy of Niti Aayog to counter or to make India Kuposhan Mukt or free of malnutrition. Now, what is interesting is to note that this particular biofortified variety is also able to give a good yield in field. And whatever claims that our scientists have been making, we have been giving these breeder seeds of these varieties to the farmer. And one such farmer, a woman farmer, who has been using this particular variety, she has been harvesting more than 10 quintals of seeds per acre of the plot. And this makes it one of the very high yielding varieties of the wheat. Another durum variety is MSCS 4058. Now, this variety is also like 4028 in the sense that it also requires very less water or irrigation. It is mainly a rain-fed the rain-fed variety. However, in case there are very few or scarce rains are there, then in that case one can irrigate this variety only twice, and with two supplementary irrigation one can get up to more than 37 quintals per hectare of the yield. And it also reduces reliance on the fertilizer usage to only 75%. And this is also very important one. Excessive use of fertilizer has been making soil saline, has been reducing the fertility of the soils, but the varieties developed by Agharkar Research Institute have been responsible mainly for reducing the reliance, reducing the use of fertilizers, and as a result, these varieties are becoming more and more popular among the farmers. Similarly, soybean varieties have also been developed by Agharkar Research Institute, and 1460 and 1520 are the two varieties which were developed by our institute. And these are also high yielding varieties. They also reduce the water requirement, but more importantly, they have short duration of less than 90 days for maturity, which is far or significantly less than otherwise the required period of 100 to 120 days. And that is an important contribution by Agarkar Research Institute. Our institute has been developing these seeds, and these breeder seeds are being shared with the farmers, and their impact on the economy has been very significant. And if I may just share it here, that the impact on the economy by the sale of breeder seeds developed by Agarkar Research Institute is to the tune of almost 2,000 crore rupees. Our institute and our scientists have also taken this as a social responsibility. And what our scientists have been doing has been interacting with the farmers in the form of arranging farmers mela or open field days, where they have been arranging the visits by the farmer. They are reaching out to the farmers, giving them information, giving them sharing with them the importance of use of ARI varieties. And as a result, more and more farmers have started using the ARI breeder seeds for cultivation of wheat and soybean. And as a result, our contribution has also been increasing dramatically. Also, our group, the genetics and plant breeding group, has taken a challenge that in this particular era where funding has been becoming scarce and scarce, they have started developing the public-private partnership. They have been interacting and they have been collaborating with the industry, and with the sponsorship of industry, they have been carrying out some of the trials. And as a result, of these trials, ARI has been generating a lot of money also, the funds also, which are again recycled back to the farming related activities. Also, our scientists have given the mini kits for trials to different farmers. This is one of the ways of making our varieties popular, and you will see what significance. On an average, our seed varieties, they yield up to 10 quintals of the yield per acre. But if you can see here, the yield has been significantly higher in fields, and some people have got as high as 20 to 24 quintals of the yield per acre. And that shows very significant productivity of our seeds, and that is why these seeds become tremendously popular. And this is again as our varieties are becoming popular. And even the Prime Minister recently released a video, and in which two of the MSCS varieties were included. Continuing with the social responsibility of our institute and our scientists, I must highlight this. This year has been a challenging year for all of us because of the COVID-19 pandemic. And this was the period when our scientists, they accepted it as a challenge. And we contributed in several ways in the fight against COVID-19. One of them has been the SARS-CoV-2 testing facility at Agarkar Research Institute. And in this particular fight against this, there was a need for Research Institute to share the burden, share the load, and undertake these trials. Our institute has developed state-of-the-art facility for RT-PCR-based diagnosis of, or RT-PCR-based testing of SARS-CoV-2, and our scientists have been doing wonderful job. The protocols were developed, they were validated, they were examined, the facility was examined by ICMR, and ultimately the recognition was accorded to our institute. And Dr. Jutika Rajwade and Dr. Yogesh Karpe, they took the lead in this entire exercise. They developed the SOPs, and as a result, the entire scientific as well as technical staff, they accepted this as a challenge, and they started carrying out the testing. And now we are able to test as many as 200 samples per day, and we have already completed more than 8,550 samples tested. But what is the most heartening feature of this entire thing is, while our scientists have been working on this, there has not been a single incidence of COVID infection being caught by any of the workers while working in the laboratory. And that shows on the expertise, that shows on the care being taken by all those who are associated with this particular testing. And I must acknowledge the tremendous efforts put by all the scientists and technical staff, and also by our purchase section, purchase department, who have been facilitating procurement of this. And I must mention here that while doing this, we have not taken any funds from anybody. We have been getting kits from ICMR, but all other consumables and everything has been from our budget, and we have been using the funds generated from industry and some part from DST funds for this particular work without affecting the research output. Our scientists during COVID-19 lockdown also were coming to the institute. They prepared the sanitizer bottles, and thousands of such bottles were shared with the Maharashtra police. And the reason for that is we really acknowledge gratefully the contribution, tremendous job done by the Maharashtra police. And we thought that this is the least that we could do. And our scientists actually did this wonderful job of preparing this. They were coming to the institute during lockdown period. They prepared this and shared it with the police. Another moment of pride for all of us has been this particular news, which appeared a few days back. US-based Stanford University has recently released a list that represents the top 2% of most cited scientists in various disciplines. Our own Dr. K.M. Paknikar, who was the director of Agarkar Research Institute and who is still actively involved in the research at Nanobiosciences, was one such scientist who was ranked in that particular list way up there. And also Professor S.B. Ogle, who is also member of the governing body of MSCS and who is emeritus professor at ICER. He was also ranked among the top 2% of the most cited scientists in various disciplines. And we are very proud that both of them are there to guide our younger scientists, and their efforts, their lifetime's achievements have been recognized in this particular way. So kudos to both of them. Our scientists have been winning a lot of awards and honors and fellowships, and this is what we have been doing. I will not go into details of this list, but I just want to include them and mention them here. So these are various awards won by our scientists at various events. Our scientists have also received fellowship from different societies, as I have included here. Our social responsibility also makes us mandatory for us to contribute in whatever way to industry and academia. We have the National Fungal Culture Collection, we have Agarkar Herbarium, and there are several other repositories that we have, which we shared for drug authentication, for sharing the microorganism, sharing fungal cultures, and so on and so forth. We are very proud to say that we have the largest fungal culture collection in India, where more than 5,000 fungal cultures are deposited. In fact, this collection has now gone up to more than 10,000 fungal species. We also have a sophisticated analytical instrumentation facilities, and various analytical services are offered to all the academicians as well as industry, and we have been generating lakhs and lakhs of rupees out of these services. We also have been organizing various courses for the benefit of students, and this has been one such area where we have generated and some funds. And what is importance of this particular fact is, during the period of COVID-19, when there was a lot of fear among all the members of the Agarkar Research Institute family, we decided to use some kind of earnings that was made out of these courses, and these earnings were used to make the life insurance or health insurance available for all the employees. Now, without taking any contribution from them during this trying period, Agarkar Research Institute offered a health insurance to the tune of 10, 15 or 20 lakhs rupees for every single employee, depending upon depending upon where exactly their level of employment. And all these employees are covered under this health scheme. And I'm proud that we have not used government funds. We have not used any contribution from the employee, but whatever funds that we earned from our activities, they were used for this particular purpose. Our scientists were also very active in publishing, and the number of publications that is published by the ARI scientists in the journals with impact factor has been every year going up slowly but steadily. The average impact factor is also increasing, And as a result, you will see that there are lots of publications that have been coming up, and we are not happy with that. We want this number to go up, and we will be taking a lot of efforts very soon to increase this number. And we hope to have a significant leap as far as the impact factor and number of publications per scientist is concerned. There are certain students who were awarded PhD, but one of the most important aspect has been this: we have to release our reliance on DST for funding. We have to generate more and more funds, and as a result, we have been encouraging all the scientists in ARI to interact with industry, generate industrial funds, and to a great extent, our institute has been successful in generating such funds. And DST also had made it mandatory for us to generate 5% of the implication towards implementation of the seventh pay commission. Now, for that, we had to share certain amount with DST, and I'm very happy to say that we have been able to generate a lot of industrial funds, and as a result, we have contributed to DST 4 times that amount than what we had committed. So that shows how our scientists have been accepting this challenge and generating a lot of industrial funds from this. Now, this is possible only because of the support that we have been getting from the stalwarts, from the members of the governing body of MSCS. And we had the governing body of MSCS, and very recently the new governing body was elected. And we are very fortunate to have the stalwarts such as Padma Vibhushan, Dr. Anil Kakodkar sir, Nitin Karmalkar sir, Vidya Gupta madam, and Dr. Ghaskar B as the office bearers, and who have been there, who are always available to guide our scientists, guide our students, motivate them, and guide them to take the right path. And in addition to that, other members of the governing body such as Professor Ogle, Dr. Ratnaswamy, Dr. Professor Jayant Pal, Dr. S.K. Apte, and Dr. Bapat sir are there. And I must say that many of these members, they visit our fields, they visit our lab routinely and help us in our research challenges. And these are the other members of the Institute Council, Research Advisory Committee, and Finance and Budget Committee, and we gratefully acknowledge their contribution. In the end, before I say final words, I want to thank all my fellow staff members, all my fellow scientists, the technical staff, the administrative staff, the students. They all have been pillars of this institute. They have contributed immensely, especially during the trying times, even when we were under the lockdown and government had made it mandatory for us to attend the office, though at the reduced strength. I must say that our staff were always more than enthusiastic to attend the office, and our staff strength was always more than whatever was required. So I must thank them and gratefully acknowledge their contribution, and the way they have been following the social distancing guidelines to fight the COVID-19 pandemic, that is commendable. So I thank them all, and whatever progress we have made is possible only because of them. I believe that there are new challenges before us, and we will be taking right step under the guidance of newly elected governing body and with the cooperation of all the fellow ARI family members. Thank you very much.
Thank you, sir, for that very crisp overview of all the achievements of the
Over to you, Kantimati madam.
And well, now we have a short film of on ARI of MACS for all of you, and Dr. G.K. Wag will be telling us more about this. Dr. Wag, please.
Dear viewers, this year Agarkar Research Institute is celebrating its 75th anniversary. At the same time, the Department of Science and Technology is celebrating its 50th anniversary. To celebrate this occasion, the Department of Science and Technology has made a short film on the research activities at Agarkar Research Institute. The film is made by Vigyan Prasar, which is an autonomous organization of the Department of Science and Technology. We are thankful to both DST and Vigyan Prasar for their kind gesture. Vigyan Prasar has shared with us the link to the film on this occasion of the birth anniversary of Professor Shankar Purushottam Agarkar, who is the founder director of this institute. We'll get to see the film in a few moments. Thank you.
The city of Pune in Maharashtra is renowned for a variety of reasons. Cultural capital of Maharashtra, education hub, IT hub, automobile hub are among the most popular identities of Pune. A lesser known fact is Pune has its first science research institute in the form of Agharkar Research Institute. Maharashtra Association for the Cultivation of Science is the parent body of Agharkar Research Institute, was established in 1946. Professor Shankar Purushottam Agharkar was one of the distinguished founder members who led the NACS Research Institute as its director between 1946 to 1960. Professor Shankar Purushottam Agharkar and his wife Parvati Bai donated all their belongings to the institute. Later, the Government of India's Department of Science and Technology (DST) placed the NACS Research Institute on the permanent list of autonomous research institutions. NACS Research Institute was renamed in 1992 as Agharkar Research Institute in memory of Professor Shankar Purushottam Agharkar.
Agarkar Research Institute was founded in 1946 as a research institute of Maharashtra Association for Cultivation of Science. Initially, the research institute started as a platform for retiring scientists who could contribute significantly to the growth of science as well as knowledge in various subjects. Ever since, Agarkar Research Institute has grown into a center of international repute under the umbrella of Department of Science and Technology.
The vision of Agharkar Research Institute is to excel as an internationally recognized center of multidisciplinary research in science and technology. Agharkar Research Institute envisages the mission to conduct basic and applied research in life and related sciences for human betterment, explore the genetic diversity of microbes, plants, and animals, develop sustainable technologies for a cleaner environment, agriculture, and better health. The research and development activities of Agharkar Research Institute span across 6 themes, namely biodiversity and paleobiology, bioenergy, bioprospecting, developmental biology, genetics and plant breeding, and nanobioscience. Biodiversity and paleobiology. Biodiversity and paleobiology group focuses on research related to living and non-living life forms ranging from microorganisms to trees. Study of micro and megafossils too is an interesting aspect of research under this theme. Collection of field specimens and maintaining them in repositories has been passionately pursued at the Institute. The NACS collection of microorganisms is recognized by the World Federation for Culture Collections. Collections of microorganisms used in biomining processes, metal and microbe interactions, and industrial waste treatment are available with the repository. Similarly, methanogenic and other anaerobic bacteria alkalophilic and thermophilic bacteria are housed in MCM. Agharkar Research Institute has a DST national facility by the name National Fungal Culture Collection of India. It is affiliated to the World Federation for Culture Collections and has accreditation of the World Data Center of Microorganisms. The institute also houses the Ajrekar Mycological Herbarium, which is internationally recognized. It houses 1,025 fungi and 40,000 lichens.
Systematic research on fungi and lichens were strengthened after the establishment of Mycological Herbarium in 1970s and was named as the Ajrekar Mycological Herbarium, that is AMH. Now holds more than 40,000 fungi, including mushrooms and lichens specimens. Fundamental research is the testimony of the fact that significant presence of fungi and lichen in all major terrestrial ecosystems make them especially valuable and exciting area of research. Considering immense potential of different groups of fungi and in recognition of expertise and resources available at this department, a national facility was established in 2008 with the financial support from Department of Science and Technology, Government of India. Under the facility, an international recognized live repository named as the National Fungal Culture Collection of India has been established in 2008 and internationally accredited as an affiliate member of the World Federation for Culture Collections.
The collection of angiosperms is maintained in the internationally recognized Agharkar Herbarium. It has more than 33,000 plant specimens from different parts of India. More than 2,000 samples of authenticated crude drug materials are also available here. Both classical and molecular taxonomy research is being carried out here. Micropropagation of rare plants is also being done. South Asia's largest repository of diatom specimens with more than 3,000 samples is also housed in the institute.
The plant and diatoms group of the biodiversity group of our institute is very unique, where we study the plants and diatoms for their taxonomy, phylogeny, DNA barcoding, ecology, and conservation using modern tools and techniques. I would like to mention that during the last 5 years, we have discovered 4 new plant species and more than 50 new diatom species from our country. And we are also making efforts to conserve many of the endemic and threatened ones via culturing them in the lab conditions and reintroducing again them to the wild. Moreover, we also render the crude drug authentication service to the researchers and general public. And our repository of plants and diatoms is also worth visiting, which holds almost 30,000 plant samples and more than 3,000 diatom samples, which is the largest collection of diatom in Southeast Asia.
Agharkar Research Institute has the largest repository of fossils in Western India. It has 5,500 mega fossil type specimens and over 2,700 microfossil type specimens. The fossils range between 1,000 million years to 12,000 years. The fossil groups include mollusca, brachiopods, annelids, fossil fish, plant fossils, arthropod, protozoa, and trace fossils.
In paleobiology group, we carry out research on fossils from different sedimentary basins and ages of the Indian Peninsula. Working on fossils helps us to understand many things about the history of the Earth, including origin of life, evolution, as well as migrations of different fauna and different periods of time. This department has a large repository, one of the biggest in India, and we have specimens, over 7,000 type specimens, both mega as well as micro, including vertebrates, invertebrates, as well as plant fossils. These fossils help us to know so many things about Earth's history, and it is our endeavor to carry out sincere research so that we can conserve our nature.
Bioenergy. Research in bioenergy spreads across generation of biogas and recovery of oil from oil wells. Frontline research on generation of methane from lignite is promising. Collaborative research with the Oil and Natural Gas Commission on microbial enhanced oil recovery has been patented. A method is developed to mitigate the problem of air pollution caused by the stubble burning of paddy residues or rice straw in Punjab and Haryana.
Our group is basically focused on developing bioenergy strategies from bio-waste. We are focused on agricultural waste. India being an agricultural land, we generate a lot of agricultural waste which is burnt and a lot of pollution is caused. To avoid that, we are developing formulations, microbial formulations, which comprise of anaerobic fungi, bacteria, and methanogens. This is used for conversion of waste to biomethane, bioethanol, and biohydrogen. Secondly, we are scaling up the process of this biogas generation. So, analyzing the process parameters for the same. Methanotrophs from rice fields have not been studied in India, and we are the first ones to study these methane oxidizers. Methane oxidizers can be used for methane mitigation studies and biotechnological applications, and we are currently looking into it.
Bioprospecting. Discovering molecules from biological resources is the main focus under bioprospecting. The development of an Android-based app for the measurement of hemoglobin for resource-poor settings has attracted the attention of pathologists. It will be interesting to see how the app works. Current areas of research in this group include semiochemicals as attractants and repellents, understanding mechanisms of metabolic and neurological disorders and cancers, development of novel treatments based on natural and synthetic molecules, development of nutraceuticals for metabolic and neurological disorders.
Bioprospecting group in Agarkar Research Institute works on discovery and commercialization of chemicals obtained from natural resources such as plant, bacteria, and microbes. We use these natural products and their synthetic derivatives for their applications in pharmaceuticals, nutrition, and agriculture industry. Understanding of mechanism of metabolic disorders, cancers, Alzheimer's is main focus of our group, especially role of copper and zinc and other micronutrients in metabolic disorders have been studied by our group for a long time. We have recently developed mobile application for estimation of hemoglobin, especially for remote areas as well as low-income settings.
Developmental biology. How can a fertilized egg give rise to a complex multicellular organism? How behaviors of cells in the developing embryo direct distinct processes. How genes contribute to the overall development. These are some of the questions to which answers are being explored in the area of developmental biology. A diverse group of model organisms like Hydra, Drosophila, and zebrafish are being used to address some of the fundamental questions in animal development. Some of the interesting topics that are currently under intense investigation are cell-to-cell communication, molecular basis of pattern formation and stress response, synapse formation at the neuromuscular junction, regulation of autophagy during development, heart development and regeneration, blood vessel development, and stem cell biology.
How can a fertilized egg give rise to a complex multicellular organism? How behaviors of cells in the developing embryo give rise to distinct cellular processes? And how genes control the overall development of the organism? These are some of the questions which we are trying to address in the developmental biology group at Agarkar Research Institute. To address these questions, we use model organisms such as Hydra, Drosophila, and zebrafish. Using these organisms, we are trying to understand some of the fundamental questions in biology.
Genetics and plant breeding. ARI plays an important role in the crop improvement program. This means improving crop productivity and profitability without sacrificing ecological and economical sustainability. ARI has all the expertise in developing high-yielding, disease-resistant soybean, wheat, and grape varieties. It is one of the leading research centers under All India Coordinated Research Projects of the Indian Council of Agricultural Research. ARI scientists have so far developed 12 wheat varieties, 8 soybean varieties, and 1 grape variety. In this way, ARI scientists are helping the farmers to boost productivity and making the country rich in food grains. MACS 4028 biofortified durum wheat variety, notified in 2019, supports the flagship program National Nutrition Strategy of the Niti Aayog Government of India, achieving Kuposhan Mukt Bharat. MACS 4028 was chosen for a UNICEF-funded farming system for nutrition project. It was operated through the Indian Council of Agricultural Researchers, Krishi Vigyan Kendras, as part of the National Nutrition Strategy. The farmers are doubling their yield by NACS 6478, a wheat variety bred by ARI. This variety gave higher value of yield, net return, and high benefit cost ratio, better nutritional quality with high protein 14%, zinc 44.1 ppm, iron 42.8 ppm, excellent chapati and bread quality. Soybean varieties with reduced anti-nutritional factors for making them more amenable for direct consumption are being developed.
The main focus of the group is on sustainable enhancement of crop productivity by developing high yieldings with disease resistance, heat and drought tolerance, having good end-use quality. ARI is also involved in breeder seed production of wheat and soybean, which is supplied to seed multiplication agencies in national seed chain. Along with this, technology transfer to farmers' field is achieved through frontline demonstrations and public-private partnerships. To complement ongoing breeding activities, biotechnological approaches are also being adopted, and molecular markers and other genomics-assisted tools are being used for efficient selection in breeding programs. ARI also has well-equipped laboratory for assessment of wheat quality.
Grape variety ARI 516 has been released for cultivation in Maharashtra, Punjab, Telangana, and Tamil Nadu due to its high yield, flavor, and processing qualities. ARI 516 is a multi-purpose grape variety with a musky flavor and is moderately resistant to fungal diseases.
We are basically working on two aspects of grapes. First aspect, collection and evaluation of grape germplasm. In this, we are having 25 species of wild grapes from family Vitaceae and Liaceae, 6 variety species, 62 cultivars, 6 wine varieties, 8 rootstock, and about 480 hybrids. Second aspect is hybridization and evaluation of hybrids. In India, grapes are mostly grown for table purpose. All commercial varieties under cultivation are susceptible to fungal diseases like downy mildew and powdery mildew. Hence, we are developing varieties with the aim to have high-yielding varieties with better quality characters and disease resistance.
Nanobioscience. The focus of research in nanobioscience has been on the development of technology in medicine, agriculture, and environment. Various aspects of study involve synthesis and characterization of nanoparticles for drug delivery, vaccine delivery, siRNA for insect control, and topical dressing for rapid hemostasis. Application of gold nanoparticles in rapid field usable diagnostics such as mycotoxin detection in food and feed, viral disease of shrimp, and prawns.
We work primarily in the applications of nanotechnology in the healthcare and agriculture. We have recently developed a hemostatic agent in the form of a gauze and a gel, which can be used in case of trauma patients as well as for stopping the bleeding in the soldiers on the battlefield. Apart from that, we have developed several kind of nanoparticles, especially the silica nanoparticles, the dendrimer kind of nanoparticles, the metal oxide nanoparticles, which can be used for several applications like drug delivery, delivery of siRNA, and other antigens, for example, in vaccines. Apart from this, we are developing gold nanoparticles based diagnostics, and they help in detection of various diseases caused by viruses in case of aquaculture. Those particles can also be used for the detection of mycotoxins in food and feed, as well as the SARS-CoV-2, the causative agent of COVID-19. Quantum dots have been developed for the intracellular imaging purposes.
Agharkar Research Institute has been on a path that strikes a balance between fundamental and applied research.
The research at ARI is carried out in 6 thematic areas. This research is a nice blend of basic, fundamental, and applied research. The quality of fundamental research is evident from the fact that ARI scientists have published more than 3,000 papers in journals of high impact. The research at Agharkar Research Institute has also resulted in developing different processes, technologies, and products. Many of these products have been commercialized by transferring the process know-how to different industries such as ONGC, KPIT, ONGC Energy Center, as well as DSM and other industries of international repute. Agharkar Research Institute scientists have collaborated with various universities as well as academic institutes in India as well as abroad, including the countries such as Japan, Russia, America, Sweden, Thailand, Mexico, etc. The doctoral research at Agharkar Research Institute is carried out under the umbrella of Savitribai Phule Pune University. Today, Agharkar Research Institute has been doing research which caters to the betterment of the society. We have decided and we will continue doing justice to the funds which have been made possible through Department of Science and Technology, and we will try to use these funds as well as industrial contribution judiciously to better the health as well as prosperity of the society.
ARI has been at the forefront of contributing in the national missions like Swasth Bharat, Swachh Bharat, Make in India, Innovate in India. The long journey of Agharkar Research Institute will enter its 75th year in October 2020. MACS Agharkar Research Institute will continue to serve the nation with its multidisciplinary. The Department of Science and Technology is completing 50 glorious years in the service of the nation through science and technology. This journey of relevance and excellence has not been a solitary one. All our autonomous organizations have played a critical role in driving forth with quality and focus and speed in their respective areas. They have excelled in their pursuits. I wish all the autonomous organizations and their scientists a very successful future. The future is coming at us at faster and faster speeds. I'm sure you will be fully prepared to meet the challenges and create new opportunities and work for an Atmanirbhar Bharat that can be proud of its S&T depth and spread.
Well, welcome back to our celebrations, and yes, I would like to thank the Vidyan Prasad as well as Dr. T.K. Var for coordinating in the filming of this particular short film. And at this juncture, I would like to welcome Professor N.R. Karmakar, who joined us a few minutes ago. And on this occasion, I have the great pleasure of introducing the respected newly elected office bearers of the NACS to you all. We are indeed blessed to have an luminary like none other than the renowned nuclear scientist Dr. Anil Kakodkar as the president of NACS. He is presently the chairman of the RDSTC, a recipient of all the three Padma Awards for his immense contributions to nuclear science, recognized globally. He is a familiar name in numerous boards of governors of various organizations where he usually dons the hat of the chairman. A member of the different Indian science academies, he is a member of the International Nuclear Energy Academy, honorary member of the World Innovation Foundation, Council of Advisors of World Nuclear Association, and many more. Welcome, sir. It's indeed our great fortune that we have with us the Honorable Vice Chancellor of the Savitribai Phule Pune University, Professor N.R. Karmakar, as our Vice President. All Punes or Maharashtra doesn't need an introduction to Professor Karmakar. He is a very able administrator as well as an educator and has introduced many reforms in the education system, both at the postgraduate level as well as the undergraduate level. Till a month ago, he was also shouldering the responsibility of the VC of the Shivaji University. He prefers to be recognized as a geologist, an igneous petrologist, delving into the depths of the mantle that has given rise to our Mother Earth. His contributions have been immense in the field of geology and recognized globally. Welcome, sir. Dr. Vidya Gupta, a plant biochemist and a molecular biologist, has focused her contributions on crop improvement and plant protection, which directly impacts our life. She has been instrumental in feeding the crores and crores of Indians. Presently, Dr. Gupta is a research advisor, ICRTC, Wagholi, Pune. She is former chair and chief scientist, Biochemical Sciences Division, CSIR NCL, emeritus scientist and professor in Biological Sciences, ACSIR. She has 4 US, 2 Indian, and 1 European Union patents to her credit. A member of several scientific bodies like ICAR, DBT, ICMR, and so on. Several awards to her credit, including the prestigious CSIR Innovation in Science and Technology for Rural Development and the CSIR Raman Research Fellowship. She has also been associated with ARI for a number of years now, and we now hope, and we are very grateful to her for having accepted the post of the Secretary of the NACS office bearers, and we look forward to her counsel and advice. Well, Dr. Surendra M. Bhaskarji really needs no introduction to anyone at ARI. He is a well-known cell, developmental and molecular biologist who has employed a variety of animal model systems in his widely acknowledged contributions. We have known him as a senior scientist and mentor all these many years. Now he dons a new hat, that of the very important position of the treasurer of MSCS. With that, I now request Dr. Daskarji to deliver a talk and addresses on his newfound capacity. Dr. Daskarji, please.
Thank you. A very good morning to all of you. Respected members of the governing body, staff, students, and scientists of ARI, and ladies and gentlemen. When I joined this institute 35 years ago, I did not have an idea that the association would be so long and would continue in more than one capacity. My friend and colleague, Dr. T.G. Baby, and I started developmental biology at Agarkar Research Institute in the mid-80s. And from then on, we have been making small contributions in the beginning and later on fairly significant contributions to developmental biology. And now we have a very vibrant group of developmental biology in MACS ARI. I will briefly tell you what developmental biology is and what are the contributions of our group. I'm going to try and make it non-technical so that everybody is with me. One of the things that developmental biologists address is how different kinds of organisms arise and how they manage to remain, how they manage to survive and reproduce together in nature. At the same time, in a particular species, as one can see here, how the same human genome can code for very different individuals. For example, Amitabh Bachchan on one hand and Mr. Sachin Tendulkar on the other. So there are these two things that developmental biologists can address easily. And one of the things that we address at Agarkar Research Institute is how mechanisms are conserved during evolution on one hand, which generates such a kind of diversity, but at the same time, how same genes and proteins function in different ways so that similar kinds of molecules can produce an insect as well as a giraffe. Developmental biology in broad terms is the study of development and growth of animals and plants, and it now encompasses all processes from fertilization to aging and death. So one can appreciate that almost everything to do with development and growth comes under developmental biology. I'll just give you a brief introduction of how we develop. Each one of us starts our development as a single cell, which is as big or as tiny as a full stop in the book. So all of us start development as a single cell. You can see the nucleus dividing at the center. And this cell divides to give rise to 2, 4, 8, and several cells. So to begin with, each one of us is just a ball of cells. And these cells later on participate in how an organism develops. So you can see that while we start development as a single ball of cells, ultimately many structures form, organs and systems develop, and by 4 weeks, by 10 weeks of gestation, we have the human fetus in place. And you can see that this is 20 weeks fetus inside the mother's womb. So from a single cell, an organism with trillions of cells, with all the organs and systems in the proper location in the body is produced. And developmental biologists generally try to find out how this actually happens. Genetic code can translate a three-dimensional organism. So one of the important things to understand development is to find out how the different structures of our body develop. And one of the important things that develop very early in the life cycle of an organism are the axes. That means head, foot, dorsal, ventral, and so on. So unless these axes are decided during early development, the various organs and systems will not develop in the proper place. For example, unless the anterior is decided, the brain will not develop at the proper place. Unless the dorsal is decided, the vertebral column will not develop on the proper place. So developmental biologists essentially try to address how these processes are regulated and what kind of cells, molecules, transcription factors, and various other epigenetic factors, that means which are not included in the genome but are environmental, participate to give rise to a three-dimensional organism. So you have the human being on the right, which is one of the most complex organisms. You have another vertebrate here, a fish. It has dorsal, ventral axis, the anterior, posterior axis. And one also has simple organisms like the one shown here, which has only one major axis, the oral axis, the oral end and the aboral end. Oral obviously refers to the mouth. So these changes are brought about in various organisms by similar kinds of proteins, and developmental biologists would try to understand how this actually happens and how it is regulated. Since it is not possible to study each and every organism that is present in nature, biologists in general tend to choose model organisms because of certain advantages they offer. These are essentially organisms which can be brought to the laboratory, can be maintained fairly easily with less funds, and at the same time, they are able to answer questions that a scientist would like to answer. So these are some of the models, for example, the worm Caenorhabditis elegans, the fly Drosophila melanogaster, a frog embryo here, a chick embryo here, a zebrafish embryo here, and a mouse embryo. Because we are mammals, we would like to study how mammalian development takes place by studying a mammalian embryo. At the same time, there are certain simple, relatively simple model systems like sponges, Dictyostelium, and Hydra. So these are the model systems that are employed by scientists depending on the kind of questions they want to answer. And every model system has several advantages and several disadvantages. And there are also some shortcomings in every model system. So choice of the model system is extremely important in answering any question. The developmental biology group at MACS focuses on embryonic development and regeneration in animals. I told you that developmental biology has very vast scope, but we concentrate only on early embryonic development and how organs and systems regenerate in various animals. So the emphasis in our group is on finding out the roles of stem cells, transcription factors. These are essentially proteins which activate transcription of genes, and signaling molecules, because most of us are metazoans or multicellular organisms. The cells in our bodies need to communicate with one another, and the cells talk to each other through these signaling molecules. We use a panoply, a combination of techniques in conventional morphological assays, as well as modern biology, in which we use confocal microscopy, recombinant DNA technology, CRISPR-Cas, and such methods. As was mentioned earlier in the film as well as in the director's report, our group uses three model systems. One is Hydra, the other is Drosophila, which is a fly, and the third is zebrafish, which is a vertebrate model system. These two on the left are invertebrates. I have three colleagues in the developmental biology group. Dr. Anuradha Ratnaparki has a neurobiology lab, and she uses Drosophila to answer questions in how nervous system develops and especially how glia develop. They have made some original contributions, as can be seen from the publications in very good journals, and they try to find out the role of MON1 and RAB7. These are the proteins which participate in early development of the synapse and the glial development. The other group is headed by Dr. Chinmay Patra. He uses zebrafish, and when he joined a few years ago, the institute was kind enough to give him full support to build a very functional and vibrant zebrafish laboratory at MACS Agarkar Research Institute. Zebrafish laboratory is on the expensive side. There has been constant support from the institute to have this facility functioning all the time. Chinmay looks at the development of the nervous system, and the other major project in his lab is regeneration of heart. So unlike many vertebrates, unlike us, zebrafish is capable of regenerating its heart, and understanding how zebrafish can do that, we can probably pick up some tricks from zebrafish and apply them to higher organisms, including us. Chinmay has also has a DBT welcome intermediate grant. The third colleague I have is Dr. Bhupendra Shrauge, and he concentrates on role of autophagy. Autophagy essentially means self-eating. So cells use this mechanism to degrade and recycle the cellular components which are no more required by the body. And these processes of autophagy have now been shown to control development, and as well as if defective autophagy happens, then several diseases arise from that. So Bhupendra has also been publishing quite a lot. So all the three colleagues have very vibrant labs, and they have a large student population. My own work over the last 15 to 20 years has been on an organism called Hydra. Before that, we used to work on vertebrate embryos, such as frog and chicken embryos, and our specific interest was in how nervous system develops. Hydra happens to be the first organism in evolution which has an organized nervous system. So we thought at that point in time that working on a Hydra might give us some clues as to how nervous system arose for the first time in animals. Nervous system is very important because that tells us you can sense the environment with it. And actually Hydra started feeding actively because it has a nervous system. Sponges, which are simpler than Hydra, cannot feed actively. They pick up food particles by phagocytosis. But since Hydra has a nervous system, it can sense the prey, it can paralyze the prey and actually put it in its mouth. So these are some Hydra in the lab, live Hydra in the lab. We have hundreds and thousands of them. This is essentially a small organism, 1 centimeter long. And you can see that on one side, it has the oral end with head and foot on the other side. It usually reproduces by asexual means, by budding. Curiously, people found out way back in the 18th century that if you take a blade and take a small cut vertically in the head of the Hydra would develop 2 heads. If you take 2 cuts, it would develop 4 heads. And therefore, it got its name from the monster in Greek mythology called Hydra. And you can see that King Hercules is trying to kill the multi-headed Hydra. Let me assure you that this organism that we work with is extremely delicate and beautiful, and there is absolutely nothing monstrous about it. But unfortunately, it has got its name from a monster. So we work on this organism, and for a developmental biologist, Hydra is a dream model. One of the major reasons is that Hydra has a tremendous capacity of regeneration. So this is an experiment done in Agarkar Research Institute. One takes a Hydra, and if you cut it into 3 pieces, the top piece, the middle piece, and the bottom piece, you can regenerates the foot piece regenerates the lost head, while the middle piece is capable of regenerating both head and foot at the proper site. Now one can imagine that if you have a piece like that, and if you follow the development to a complete Hydra from here to here, at morphological, cellular, molecular, whatever level, then you can study how pattern formation takes place, how tissues are formed, how lost structures are regenerated, what happens at the level of the cells, how they talk to each other, which kind of genes are activated, what are the transcription factors which are required for cell communication. So we use this model system to understand pattern formation and regeneration. Regeneration is a very complex process, and Hydra is one of the organisms which has the simplest kind of regeneration. So you can cut a Hydra into pieces and it will quickly regenerate the lost structures without undergoing complex mechanisms like cell division or de-differentiation, de-differentiation, blastema formation, and so on. So what happens is, as more and more complex organisms evolved. For example, this is Hydra, this is Planaria, and this is limb of an amphibian, of a frog. So as evolution gave rise to more complex organisms, the regeneration also became more complex. It started involving more steps. Unfortunately, in our case, we can regenerate very few organs. We can regenerate liver and pancreas to some extent. And of course, the skin is shed off every day and so on. But we cannot regenerate other organs. And developmental biologists would like to, understand how these various organisms regenerate and whether we can pick up tricks from these organisms and can regenerate organs at least in vitro, at least in the laboratory. In addition to this, such knowledge on the basic of how organs develop and how they become functional can also help, and in fact, it is helping today to make artificial organs. For example, artificial kidneys and pancreas are being made based on basic fundamental knowledge of how regeneration takes place in different kinds of organisms. Hydrobiology at MSc ARI started, as I said, 20 years ago. There used to be a scientist, used to be a professor in Jawaharlal Nehru University, Professor Shivtosh Mukherjee, who used to work on Hydra and sponges in the 60s and the 70s. For some reason, unfortunately, this model system was not used by anybody over the last 20 years, from 1975 roughly to about the year 2000. And because we were looking at how nervous system develops, and since Hydra is the first organism to show an organized nervous system, we thought of using this system. Unfortunately, there were no other labs at that point in time in India which work on hydrobiology. So it was a little bit difficult to start everything on our own. But we had extensive collaboration with people in Germany who work on Hydra, and with their help, we could ultimately establish a functional Hydra lab. We have been looking at various different aspects of hydrobiology here. We look at genes in Hydra, which are very similar to genes in vertebrate embryos. We look at how UV irradiation affects pattern formation, how UV also damages DNA and how it is repaired in Hydra. We look at autophagy in Hydra, and we also have been looking at Hydra microbiome. All of us are associated with a large pool of microbes, and many of our physiological and apparently even the psychiatric processes depend on the kind of microbes we harbor in our body. And Hydra also has very specific microbes, and therefore it obviously, along with others, we have shown that the association between microbes and metazoan animals has started several years ago. Hydra, by the way, is evolutionarily very old. It is believed to have evolved 60 million years ago. And as compared to that, Homo sapiens, humans, evolved about 1.8 million years ago. So you can imagine how old this organism is. And in spite of that, our study and study from other laboratories show that it has very similar genes and proteins in Hydra. So over the years, what we did was we took an approach wherein we started looking for molecules homologous with vertebrate genes, which are important in development. We tried to find out whether they are present in Hydra. So Hydra genome was published in 2010, but before that we used EST databases and so on to find out whether similar molecules are present in Hydra. And we actually ended up discovering quite a few molecules for the first time from this organism. And these molecules are of two types. One set is from the pattern formation, they participate in pattern formation, and the other set, these genes participate in DNA repair. I would like to mention here that this is the only lab in the world which addresses DNA repair in Hydra. So these are some of the molecules that I have listed. I'm not going to go into details, but noggin, for example, by the way, the meaning of noggin in English is a person's head. So noggin is responsible for our head formation during early development. It's a very important molecule. It's very critical during early development. And we found that noggin is also present in Hydra, which doesn't have a head similar to our head. But in spite of that, Hydra has these molecules. As was mentioned in the director's report, we have also found out vascular endothelial growth factor and FGF from Hydra. Now, please remember that VEGF is required for blood vessel formation in higher organisms. However, Hydra is a diploblast. That means it has only ectoderm and endoderm, and it doesn't have blood because blood is made up of mesoderm. In spite of that, it has such molecule, which means that such molecules evolved early in evolution for some different functions and have been later recruited for very different functions. So our studies can give insights into a new branch of biology, into a new branch of developmental biology, which is known as evolutionary developmental biology, or EVO-DEVO for short. So these molecules and their structural and functional characterization offers us insights into how evolutionary developmental mechanisms evolved. And that gives us an insight as to how similar kinds of molecules on one hand can make a hydra, on the other hand can make a monkey or a giraffe or a human being. So these mechanisms of formation of three-dimensional organisms have been conserved during evolution. As I mentioned earlier, we have also, ours is a unique laboratory in the world which looks at DNA repair genes. Xeroderma pigmentosum is a disease in humans, and these people tend to get cancer due to exposure to UV. And surprisingly, we found that all the XP genes, all the Xeroderma pigmentosum genes are present in Hydra, and they actually participate in repair of the DNA. In addition to that, recently we have discovered more DNA repair genes from Hydra. So the take-home message from our work and from others is that very similar and often identical proteins are found in Hydra, fish, frog, snake, chicken, any kind of animal that you see. And they carry out sometimes additional or different functions across different species. For example, Hydra has an insulin-like molecule, but Hydra doesn't have blood. It doesn't function as a hormone there. It functions as a cell-cell communication molecule. But insulin, in case of us, as all of us know, is very important in controlling blood sugar levels. So same molecules have been used for different purposes. Now, if you employ the same molecules for different purposes, you end up getting different kinds of animals. And that is the basis of how similar regulatory mechanisms and molecules can generate such biodiversity. And this work also shows that the basic so-called genetic toolkit of molecules evolved millions of years ago, because I told you that this is a 30 million old organism and has been refined over time. And as I said, it is one reason how biodiversity is generated. Now, to prove biological evolution, there are only two ways of proving it. And there are, as you know, there are many skeptics who still don't believe in biological evolution. However, the two ways of proving it is, one is fossil records. And we do get fossils, and we know that fossils tell us as to how animals evolved. However, fossils are very rare to find, and we don't find fossils which establish a chain of events. For example, you don't find a fossil which will tell you how reptiles arose, for example, from aquatic animals. So unless you get a clear-cut chain of fossils, it is not possible to prove that. Therefore, people use molecular biology, and with molecular biology, recombinant technology, and bioinformatics, you can now compare nucleotide and amino acid sequences in different organisms, and that clearly shows us that we are all related. So we are not all that different, not only from monkeys, but even from flies or worms. So our contribution in brief to developmental and evolutionary biology has been deciphering how cells communicate with one another during pattern formation and regeneration. And these studies provide insights into evolution of cell signaling pathways in metazones, how damage to DNA is repaired in Hydra, providing insights into evolution of DNA repair mechanisms in metazones. We have not stopped at doing our own work, but since there have been problems with using animals in practicals and research, we have introduced Hydra not only in research, but also in teaching in India. So several colleges and universities, at least in Maharashtra, have introduced Hydra practicals in their curriculum. It is easier to get permission to use Hydra than, for example, permission to use a mouse or a frog. And as a service to these laboratories, we have developed a Hydra kit. There is only one other kit available in the world, which is commercial and which is very expensive. But this kit of this kind has been introduced by Agarkar Research Institute for the first time, and we supply it without profit at a very low cost. And we have supplied more than 25 kits all over India, and people have started Hydra labs with that. Some people use these kits only to do practicals and then they discontinue. But some laboratories, for example, as an offshoot of this work, ARI Pune and University of Hyderabad have very robust hydrobiology programs. So we have been instrumental in introducing this so-called simple model system in teaching and research in India. Over the years, many developmental biologists from all over the world have visited us, and this is just one example where in 2008, Professor John Gurdon and Dr. Thomas Bosch visited us, and Professor John Gurdon, as many of you may be aware, got a Nobel Prize in Medicine in 2012 for his work which he did 60 years back, wherein he cloned an entire organism for the first time. He cloned a frog. And John Gurdon is, we use his textbooks, and it took quite a while, but finally he got a Nobel Prize in medicine. Apart from Professor Gurdon, who also examined the thesis from our institute, several scientists have visited the lab. I'm mentioning this because of our contribution, people do visit. They don't only go to Bangalore and Hyderabad, they also come to Pune. As another offshoot of our work in developmental biology, I was recently invited to guest edit a special issue of International Journal of Developmental Biology, along with Professor Nanjundaya of Indian Institute of Science in Bangalore, about developmental biology in India. So we have worked on it for 2 years. Now the issue is out. It contains about 37 articles, which tells what went on and what is going on in the developmental biology field in India over the last 100 years. So it has historical accounts, it has review articles, and it has original papers. So up till now, we have documented what has happened in this area in developmental biology. In being invited as a guest editor for this special issue is a recognition for work that is done MSCS ARI. These are the people on the left who are from my lab who have contributed to this work. The funding for this has come from Department of Science and Technology, Department of Biotechnology, ACRB, CSIR, and of course, generous intramural funding from Agarkar Research Institute throughout the last 3.5 decades that I've been working here. This is the erstwhile developmental biology group when I retired about 4.5 years ago. And whatever we could do is because of all these people. So I'll stop here, and I have just tried to give you a gist of what we do in developmental biology. Thank you.
Thank you, sir, for that very insightful talk on developmental biology. And here I now have the pleasure of inviting Dr. Vidya Gupta, Secretary MSCS, to deliver her talk. Dr. Vidya Gupta, please.
Thank you, Dr. Kantimani. Honorable Padma Vibhushan, Dr. Anil Kakotkar, sir, respected Vice Chancellor of Pune University, Dr. Karmalkar, Dr. Prashant Dhakefalkar, Director of this institute, all the senior members of MSCS fraternity, as well as Agarkar Research Institute, and the staff, scientists, and students. Let me first congratulate the present director as well as the earlier directors of ARI for bringing this institute to such an height that we heard from the presentations of Dr. Dhakefalkar as well as Dr. Ghastadvi and the film. I feel really honored to be with and to work under the able guidance of stalwart Dr. Kakotkar sir for fulfilling the objectives of Maharashtra Association for the Cultivation of Sciences. I have been associated with MSCS for last 13 years as life member and 5 years as governing body member. However, this is my first occasion or opportunity to be present for the celebrations of the Foundation Day of Agarkar Research Institute. I am really thrilled to hear the progress of the institute. ARI has no doubt a very great personality, Dr. S.P. Agarkar, as its founder member. He founded this institute in 1946 and remained as a founder director till 1960, till his end. So today, ARI is entering into the 75th year, completing 74 years. So we begin actually the Diamond Jubilee celebrations of our existence in scientific field in Pune and the country. So we all together, we will plan for the scientific celebrations for this entire year. Dr. S.P. Agarkar was a botanist. More specifically, he was a plant morphologist. He was interested in the genetic diversity that is existing in the Western Ghat regions. So in the present scientific programs of ARI, this particular area, the biodiversity and bioprospection, these two areas have become the niche area of research. Apart from biodiversity and bioprospection, genetics, plant breeding, microbiology or bioenergy, and other nanobiosciences, developmental biology are also the important areas of research of this institute. More importantly, the repositories, the culture collections, and the herbaria, either of plants or mycological herbaria, they are very unique features of this institute, which give real good services to the scientific community, which is very much needed in the present day science. Based on my own research and my own research interests in plants and plant molecular biology, I'm very happy to say that I have my own research interests matching with at least 3 different areas, the biodiversity, bioprospection, and genetics and plant breeding research areas of ARI directly. In fact, I have actively collaborated with the group of genetics and plant breeding, which was led by Dr. V.S.P. Rao at that time. And this collaboration was for almost 15 years. We had All India Wheat program supported by the Department of Biotechnology, and Indo-Swiss and Indo-Australian programs for wheat improvement. I feel very proud to say that our wheat molecular biology collaborative program supported by the Department of Biotechnology was the first program in the year 1995 with such a large funding, and the national program which was supported by the government for the first time. And this program continued for 10 consecutive years. And after that, of course, we had many projects on wheat rust and other wheat problems supported by DST and DBT as well as the international agencies. In that program, we joined our hands and shared expertise in wheat breeding, pathology, and molecular biology to establish foundation of molecular wheat breeding in the country. If we look at the agricultural history of independent India, we can divide it into 3 phases. The first one is a pre-green revolution period, say from 47 to 60-65, and the crop production was mostly dependent on the cultivable land, seed bed preparations, planting practices, weed control, and so on. And there was very little increase in the crop productivity that was observed in this era. But the second phase was more for 25 to 30 years, which was from 60-65 to 1995. And this was the post-Green Revolution scenario. In this period, the agriculture became more of a science-based agriculture. And the Asian Green Revolution under the leadership of Dr. Swaminathan, M.S. Swaminathan, became a modern hybrid seed, chemical fertilizers, and weed control technology based agriculture. Better and very high yielding varieties were produced with better crop management during this period. And this revolution focused more on wheat and rice as the staple foods. However, there were certain disparities during this period, the regional disparities, interstate disparities towards the inputs and yield plateaus were achieved in this period. Moreover, important crops such as legumes and oilseeds and horticultural crops, they remained neglected in this period. So although the high yielding varieties were produced, the food security issue remained kind of a burning issue in spite of high yielding varieties. Then came the third phase in Indian agriculture, which continued from say 95 till now, which is the era of agricultural biotechnology, a gene revolution. The molecular marker based breeding for high yielding varieties has become a basic slogan in this era, which provided no doubt the sweet fruits to Indian community. And Agarkar Research Institute, NCL, and many other institutes in the country, they contributed to this mission. The basic objectives of agricultural biotechnology now have been to increase the crop productivity by conserving biodiversity and using external inputs very, very efficiently, and responding to the climatic changes also very efficiently on the other hand. If we look at the statistics, agricultural statistics in the year 2019, which has been published by the Ministry of Agriculture, the trends have been set in such a way that the urban population will increase from 35% right now, approximately 35% right now in the year 2020, to almost 40% in the year 2032, indicating that there would be less agricultural labor availability, less agricultural inputs. But the food grain demand will increase to 120 million tons by 2030. But interestingly, there is also an estimation that the yields also would be very, very high. The supply would be about 125 million tons, which makes it surplus to surplus food grains for the Indian population. So we will have good surplus food grains except for the edible oil. Hence, what we need to look at is there would be a more demand for the quality food with high nutritional value. And this clearly gives us the guidelines to our future research activities as enhancement of nutritional quality as well as other quality traits in the food grains based on such statistical informations. This will help us to formulate our research programs in the coming future. I'm sure at ARI, we are ready to take up these challenges in agricultural biotechnology area. Our wheat, soybean, and grape varieties have received recognition from Indian government, no doubt. And this can now be utilized further for the higher nutritional quality traits in coming years. But this challenging task can be tackled if we join hands from various theme areas within ARI as well as from the other laboratories. We can have the flagship programs, research programs, say 2 or 3 for ARI, where such niche or need-based requirements, those can be fulfilled by various groups working together. The biodiversity, bioprospection, microbiology, nanobiotechnology groups, they can join hands with genetics and plant breeding group to undertake such massive effort of flagship research at ARI. I am sure we can establish more such programs and research activities where many ARI scientists, they can work together on really important projects which will give ARI a very good name and a better visibility in coming future. And we can initiate such activities as our mission mode program or mission mode activity in this Diamond Jubilee year of ARI. Thank you.
Thank you, ma'am, for that very encouraging words about the work being done at ARI and also your advice and guidance to all of us. Now, I wish to call upon our Honorable VC, Professor N.R. Karmakar, to address all of us.
Very good morning. Indeed, it's a pleasure to be with you all on this very auspicious day, the Founders Day. Honorable Anilji Kakodkar, the President of the present governing body, Vidya Ji, then Gas Kadbi, the present director Prashant Dhakipalkar, rest all senior scientists, and whoever has joined in online mode. It's indeed a pleasure. I'm very sorry that I could not join on time. The reason being a month before actually the MOU signing was decided, and this happened to be at the same time at 10 o'clock in the morning. Since we are launching basically a program in jewelry designing and gemology with Indian Institute of Jewelry and Gemology, Mumbai, and those people had come, and therefore I could not join on time. But last year, I remember last year, on the same day, in fact, I had given a more of a technical sort of a lecture on mantle petrology, and that is what is the subject I love the most. It's my passion. And when you must have learned it from Dr. Kantimati that I am a geology person. So more or less always enjoys more in the past rather than basically living things like biology. This is more of a biological institute, and which was established in 1946 as Maharashtra Association for Cultivation of Science and Research, and then renamed in 1992 as the Agarkar Research Institute. My association with this institute in the capacity of geology basically was with the paleontology department, which to an extent did wonderful work under the leadership of C.W. Chiplankar, who in fact even taught us not only paleontology but the engineering geology when I was doing my master's in the campus of the University of Pune. And that is what perhaps is the smallest association that I have with this institute as such. Presently, I am also looking after, in the capacity of the chairman of a governing body, another institute, that is Birbal Sahni Institute of Paleosciences, the previously Paleobotany Institute as such. So wherein again it is a biology, and surprisingly, I'm a geology person. But anyway, when I'm getting associated with this institute, which is a very renowned institute in the field of biology, which includes basically plant science, then microbial, and in recent time, in fact, the life science also, plant science and life science together. And there are various different disciplines that wherein a very wonderful kind of research that has been happening. I had the fortune to listen to Gas Kadbi, although I missed out the film on MSCS, but the lecture delivered by Gas Kadbi sir on developmental biology as such. And biology initially under the umbrella of life sciences, there used to be only few departments like Department Botany, Zoology, and perhaps during my time, another branch that came in microbiology. But now it has moved on to bioinformatics, biotechnology, and the molecular biology, and so many minute sort of a specialization, micro specialization that has come into play. And especially the importance of these disciplines we understood the most during the pandemic time, which began sometime in the month of March. Since then, everything is happening in an online mode. There is hardly any physical sort of interaction that has been happening. And today, again, we are in online mode actually discussing about the developmental biology and rest of the disciplines as such. So, as rightly been pointed out by Vidyaji, that there is a tremendous scope in expanding in various different micro specialization, micro areas as such. Although at the same time, we shouldn't forget about the past, like the paleontology or paleobotany. Those should also remain basically part and parcel of our research domain as such. And that is what I would like to emphasize the most here. Of course, this has been happening basically, perhaps this is the second meeting after taking over charge of the vice presidentship, and perhaps the actual working is yet to begin. But the past experience that I gathered from Birbal Science in the Sani Institute, which is also an autonomous DST institute funded by DST, and wherein obviously there are various different domain that needs to be looked at, especially in the governing body. For example, the RC Research Council, wherein most of the research scientists expected to submit small proposals and which get funded. And that is how basically the progress and the areas get defined as such. That needs to be actually expedite. We have to actually work on it at the immediate in the immediate future, I would call it like, and that is what is required the most to give the boost for this research activity in this particular institute as such. When we speak about basically my own discipline, that is geology, although it is related to rock and fossil record as such, but that is what is very important, because this is the only planet which is a living planet. And it is living only because basically the interior wherein I specialize, basically interior of the earth is hot. The engine is running only because it is charged with fuel as such, and there is a plate tectonics, the movement of plates, and there is a seamless interaction that is happening between the lithosphere, then biosphere, then atmosphere, and perhaps hydrosphere. All these four spheres exist only in this planet Earth as such, and that is the reason it is a living planet. And therefore, one of the verticals, obviously the biosphere, which to an extent this particular institute is specialized in. And that is the area wherein we need to expand further. We have to have handholding with many of these similar institutes, because all these days we have worked more in silos as such. When I took over as a vice chancellor of this university, in fact, that is what the dream that I had, that there has to be a seamless sort of interaction happening among the various different disciplines as such, which wasn't happening previously. Everyone basically was working in silos, confined to their own department. So that kind of a culture we need to break, and there has to be interaction happening, because this is the time wherein we are speaking about multidisciplinarity or interdisciplinarity, and which to an extent would pave the way ahead. And that is what I'm looking forward even in this particular institute also. On the campus of the University of Pune, we have the Cell Biology Institute, the NCCS. Then apart from that, there are 6 different branches of biology that we host on the campus, whether it is Institute of Bioinformatics and Biotechnology, IBB, or Department of Biotechnology, or Department of Bioinformatics, Microbiology. So the campus department, the campus institute, and similar institute in and around. Even the ICER to a great extent is doing wonderful work in this particular area. So unless and until we do a handholding and maybe a combined sort of a project together, perhaps that is the way basically taking discipline side and the research ahead as such. And biology is in demand, especially at the backdrop of the present pandemic as such. And we have learned actually even in the developed countries also, they couldn't cope up with this particular pandemic. We are virtually vulnerable. So there are areas, niche areas, which we haven't yet understood as such. We haven't even got what is called basically the vaccine. We've been speaking about it as such. But there are institutes like the Serum Institute located in this very city as such. And we have to have a very strong sort of interaction with such institutes. So there has to be a sort of a multidisciplinary approach, and that is how basically we intend to, or rather that is what is my intention in looking at this particular institute, basically doing many projects together with the allied institute or allied research institute as such. And there has to be somewhere, because all these days we've been more talking about basically doing research and publishing papers in good index journals, such. But beyond that, we have to look at whether it is patenting or whether it is commercializing the patents as such, developing the prototypes, and then basically scaling it up further. And that is what, in fact, the DST is also looking at. In one of the meeting, in fact, the secretary made it amply clear, sound and clear, that institute actually should take efforts to make it a sort of a sustainable institute. They have to produce their own funding. So that is what basically the entrepreneurship development, that is the area wherein we have to strengthen further. So these are some of the things that we would be discussing perhaps in the regular governing body meeting as such. And I'm very happy that I'm closely getting associated with this institute, and through basically a sort of a consolidated effort with the institute, with the university, with the allied basically institutions in and around Pune, or maybe beyond Pune, we would like to actually again take further the activities of the Agarkar Research Institute as such. So I wish every one of you a very happy future, a wonderful future in this institute, as well as a good future for this institute. I'm thankful that I've been given a chance to make my mind clear during this particular period. Thank you once again. I wish you all the best, and Diwali greetings to everyone. Thank you so very much.
Thank you, Professor Karmakar, for your vision for our institute and for our well-being. We now move on to the second half of today's Founders Day function. In an effort to curtail government expenditure, the Government of India has suggested many measures. One among them is the printing of books. However, to ensure that scientific developments reach out to a larger audience for the benefit of the society, systematic compilation of information, know-how, etc. is essential. So, following the directives of the Government of India, our scientists have come up with 2 e-books. I now invite the authors, Dr. N.M. Datta and Dr. R.K. Chaudhary, consecutively, to tell us about the rationale behind these publications. Dr. Datta, please.
So, very good morning to everybody. Thank you, madam. Very good morning to everybody. We are extremely delighted that the book Compendium of Raw Botanical Drugs of Maharashtra, authored by Dr. Anuradha Upadhyay and me, is getting released at the hands of Dr. Anil Kakodkar, who is president of MACS. And this release will be in presence of eminent personalities like Dr. Karmalkar, Dr. Vidya Gupta, Dr. Ghaskadvi, and Dr. Dhakevalkar. Dr. Kakodkar, incidentally, is also a chairman of Rajiv Gandhi Science and Technology Commission, Mumbai, which is the sponsoring agency of this particular book. The book is an outcome of this RGSTC funded project of developing a botanical drug repository, and the project was operated at ARI from 2014. Since adulteration and substitutions are major issues in raw botanical drugs, the exact identity of plant parts which you use as a crude drug or raw botanical drug is a very important component. So there is some sporadic literature which deals with identification of plant parts that are used as crude drug, a pictorial sort of representation or a pictorial sort of guide was missing. And probably this book which we are bringing up today deals with this lacunae, and it provides pictorial representation of crude drug parts of close to 200 plant parts that are used in herbal drug manufacturing. We hope that this 400-page compendium will be helpful to medicinal plant collectors, traders, pharma industries, and students who seek to identify precisely what drugs, what botanical drugs or crude drugs they are dealing with. So thank you one and all, and over to Dr. Ritesh Kumar Chaudhary to tell us about the second book.
A very good afternoon, everyone. I'm extremely delighted by the online presence of this elite gathering. I would like to thank Kakodkar sir also for taking time off from his busy schedule and spend some time for releasing our book. So at this juncture, you must be wondering about the generic name Caparis and might be willing to know more about it. I'm sure everyone present here is aware of the popular drug called Leaf-52. This is used to treat liver ailments, and just to inform you that this is one of the plant Caparis, one of the major ingredients of this drug is a Caparis species called Caparis spinosa. And in India, you can find almost 35 taxa of this genus. And believe me, they all look similar in the field, and it's not easy to identify them at the species level. And due to this difficulty in identification, they are heavily adulterated in various Ayurvedic formulations. To overcome this problem, we have come up with this book as per the instructions and funding support received from Science and Engineering Research Board, SERB, DST. So our book provides a detailed account of 35 taxa belonging to genus Caparis in India, along with their taxonomic descriptions, photo plates, herbarium images, distribution maps, along with their local names in major Indian languages. And the best part of it, the ebook will be freely downloadable from our institute's website. We hope it will serve as a great reference source and be useful to the taxonomists, pharmaceutical companies, conservationists, forest officials, nature enthusiasts, teachers, and of course, students. Thank you very much.
Thank you, Dr. Ritesh. Well, now I have the honor and pleasure of inviting Padma Vibhushan Dr. Anil Kakodkar to officially announce the release of both these books.
I am extremely happy to announce the release of both these books, one related to medicinal plant database and the other related to, I forget the name, but related to the drug Leaf-52. Congratulations to authors, the institute, and everyone else connected with bringing out these documents.
Thank you so much, sir. I shall be just sharing the screen so that all of us can have a look at how these books actually look like. That's the names of the authors you can have, Anuradha Upadhyay and Mandar Datta as the cover page. This is how it will be appearing. A snapshot of a few pages. And that's about it. Both. We have the next one. So that's where you can download the ebook freely on the Venus Catalyst. May I now have the pleasure of inviting Honorable Dr. Anil Kapoorkar to deliver his presidential remarks.
Thank you, Dr. Kantimati Kulkarni. Professor Karmalkar, Dr. Mrs. Vidya Gupta, Dr. Ghas Kadbi, all other members of the governing body, the new governing body as well as the old governing body, those who are present here. Dr. Dhakefalkar, the director of the institute, Dr. Kantimati Kulkarni, Dr. Datar, Dr. Chaudhary, all members who are present in this particular meeting, colleagues from ARI, ladies and gentlemen. Well, first of all, let me use this occasion of the Founders Day of Agarkar Research Institute to pay homage to our founder, Professor Agarkar. It just so happens that about a week or 10 days back, I came across a paper which was talking about the evolution, or I forget the title, but it was something to do with how the three science academies in the country evolved. And it's a very interesting historical account. And what was very noticeable was Professor Adharkar's name appeared there again and again and again. So he had played a big role in shaping of the scientific world in India, as it were. And of course, he did the same thing after he came to Pune. And Pune had several, Pune has been a hub of education. Several education institutions were set up. And then they realized that you also need the institutions of advanced studies, institutions of research. And so several of such institutes came in, but mostly in the area of liberal arts. And so, as was mentioned in that film, Agarkar Institute was the probably the first institute dedicated to scientific research. And we all know it began with very humble beginnings and with great support from several leading individuals as well as institutions in Pune, that this institute came up. So I think on this day, Founders Day, we should recognize all early founders, including Professor Agarkar, but also everybody else who were involved in the foundation or founding of this institute. Fast forward, I think I want to also compliment all of the present generation people associated with the institute as the institute enters 75th year. And as was said by Professor Karmalkar, I think we should certainly be planning a rich scientific program spread throughout the year uh, to kind of celebrate and commemorate this particular occasion, 75th year of the institute, culminating in a major function when the institute actually completes the 75 years. So friends, we clearly have a great legacy to take forward. You take any dimension which kind of goes to kind of make the recognition of a research institute, and you will find that Agarkar Institute mirrors well on all those dimensions, and basic research, fundamental research is at the heart of the institute. But at the same time, we have seen several applications during presentations. In fact, several presentations, 2, 3, and everywhere, including that film. This translation part was also very, very visible. So, so I think I feel particularly very fortunate to be associated with this institute, with all of you. Of course, this is not the first time that I'm interacting with the institute. As was mentioned, as a part of this medicinal plant database, which a major project that the institute ran for several years, and I think great work has been done. This documentation that we just saw released in the form of an ebook, but it's also there in the form of a hard copy. It's also there as a database, which is also on the website of the concerned departments in Government of Maharashtra, and of course on the website of ARI and the Rajiv Gandhi Science and Technology Commission. The idea is it should be available for research, but at the same time, it should be controlled from exploitation, because several valuable plant species which are important, some of them have actually extinct, and it is important that while we have a full database, but we also kind of make sure that we are making full use of these, but in a sustainable way. So there must be that element of sustained activity to propagate wherever the larger application is there, and make sure that the species do not get threatened or do not become extinct. I just want to comment at this stage on the two books that we just released. Of course, I've been quite familiar with the medicinal plant database work of Agarkar. I have come to ARI a number of times in the context of review of that work. But talking about this Sapphirus spinosa, the plant which goes primarily in this drug, Leaf 52. Now, I somehow realized about many years ago, maybe 30, 40 years ago, that Leaf 52 is more popular in Russia than it is in India. And I think it was a major export item for, if I mistake not, Jandu Pharmaceuticals at that time. So the short point is, or for that matter, in the context of the wheat varieties or the soybean varieties, it was mentioned that ARI's work is contributing to something like 2,000 crore contribution to economy every year. So clearly the work, although our focus is basic research, but this work has a lot of economic value. And more importantly, the culture that exists in the institute has enabled going deep in new knowledge creation, as was mentioned in that film, to taking that knowledge for the societal good. And I want to congratulate uh, uh, all colleagues from the institute, both present and past, to make that happen. And I think it should be our endeavor to only further enrich that tradition and take the institution to a greater level of standing. It already has a great standing, but to a much greater level of standing. Now, I want to make a few comments in that context, and I'm very glad that whatever I'm going to say is entirely in sync with what was said by Dr. Vidya Gupta, as well as what was said by Professor Nitin Karmalkar. So I think I'm very happy that uh, uh, we have kind of like-minded people within our governing body, and I'm sure all other members of the governing body and the senior colleagues from ARI will all create a group of like like-minded bodies in terms of the direction to take in years to come. Now, one of the challenge, we are turning 75, and one of the challenge of maintaining institutional excellence is about learning to maintain excellence in steady state. We have several new institutions, and when institutions are new, you are in an expanding universe. You are expanding infrastructure, you are expanding the community, scientific community, and you have a kind of well spelled out mission where you want to go as a part of institute development. And well, even there, one has to be very careful and work hard to ensure high level of institutional excellence. But I think it becomes even harder when you want to carry on the activities of the institution in a steady state. And certainly at ARI, we are in a steady state. So we may have, and I think we should have ambition to go forward, look to the future, be more ambitious. But we have to do this in steady state because, financial limitation is one. But that, I suppose, depending on the quality of work we do, we'll be able to attract more and more finance. But there are physical limitations, instance in terms of the laboratory space, in terms of the number of people that one would want to hire, although there may be need for more. And the question is, how does one maintain a high level of excellence under the so-called constraints of operating in a steady state? And I'm not going to kind of take too much of time in elaborating these points, but I want to just flag this point that we must kind of strengthen our ways to kind of maintain high level of excellence, and in fact take it to higher level while operating in steady state. And when we discuss and when we meet, we can certainly talk about this in greater detail. Another point that I wish to make is related to, again, it's related to excellence, but you get known uh, by way of the importance or the impact on problems you solve. And this is true at the level of individual researcher. This is true at the level of institution. And as I said, if your ambition is bigger than institution, because we have to work in steady state, then this is also true about the important problem that you can solve collaborating with other institutions and other individuals. So, but this definition of this problem, to be able to make larger and larger impact, has to, of course, be moved to higher and higher levels. And I want to give just some examples in this regard. For example, we have 6 thematic areas, broad thematic areas, which Dr. Lakefolkar described very efficiently about the work, and we got a full picture of the program of the institute. Now, and of course, in each one of them, some important problems have been handled. There have been applications. There have been impact on the society. We've been also able to generate a lot of collaboration with industry. And so in that context, I was just thinking, let's say on the foundation day today, I'm not suggesting that we take it up, but what are those examples of, if I can use a colloquial term, big ticket items or big ticket problems, which will make a large impact? Now, and I'm saying this because I have some background of ARI, because apart from Rajiv Gandhi Science and Technology Commission, that project where I have been coming to ARI, I am also associated with many other institutes, and where through that association I have come to know about the work of ARI. For example, mention was made about the ONGC Energy Center, OEC, and there, for example, enhancing the methane output from coal fields. And there's some good work happening which ARI is carrying out. Hydrogen mobility was mentioned about it, and I came to know about it from KPIT, and so much and so forth. So I am very familiar with the capabilities of ARI. So whatever I'm saying, please take it in that spirit, that fully recognizing that we must maintain excellence, fully recognizing that you can maintain excellence only on a very solid and strong bedrock of excellence in basic science and fundamental research. But the real impact comes on the basis of how you impact the society and the country. So, for example, biodiversity is one of the thrust area, key thematic areas for it. Now, we all know that, for example, our, environment has become a big issue. Now, today, environment, on one side, you have the environmental activists who see problem in everything that happens. Then in between, you have researchers like our colleagues in ARI or academic environment and all, and they have done tremendous work in environmental sciences. But then there is the commercial, industrial, societal domain, call it agriculture, call it other industrial activities which affect the environment. And the whole world is talking about climate change at the global level. So it's a problem at local, regional, global level. Climate change has become a life and death issue. In the sense, people are saying that unless you go to the CO2 neutral, make zero emission by middle of the century, 2050 or thereabouts, you will have warming more than 1.5 degrees. and that will have serious consequences to existence of a large part of humanity. So, we're so close to the cliff edge. I'm assuming that all the predictions, and they are of course done by large groups in collaboration. So I believe that they are true. There are evidence that those are true. But the point I'm making is, at ARI, we have work going on paleobiology. Now, because of this, call it development, call it technology or something, the changes in environment are happening much faster today than any time before. So, for example, when you come to environmental testing on materials, you have a method of accelerated tests following Arrhenius principles or whatever, all those things, I don't have to go in detail. But you can work out whether a particular material or particular device will survive the performance over expected life, 50 years, 70 years, 100 years, or whatever. And you do that in a test which is maybe a few months or maybe an year or things like that. So there are modes of looking at this in an accelerated manner. So the thought that comes to my mind is leveraging our knowledge in paleobiology, where we know how things have evolved over a long period of time. I'm conscious that there are gaps and things like that, and I'm not saying that we should get these answers today, but can we create a bit of predictive capability at least, because we know that the environmental related damage is only accelerating, and it has accelerated over a period of time. So whatever has happened, let us say over a period of 1,000 years, everybody says will happen, at least in technology sense, people do, or that Moore's law, that, so similar thing may be existing here, that whatever has happened over 1,000 years may happen in next 100 years or next 50 years or something. The challenge is, can we do a bit of prediction? And I want to take one step further. For example, the entire our food related activity, agriculture activity, there are several inputs that are costed, that gets into production, and then there is always this crisis. Farmer gets very little, consumer gets at a very expensive thing, and people are not sure whether at different steps in the value addition chain, whether the cost increase is justifiable or there is an unjustified increase. Probably latter is the case. Even after that, one finds that there are several very important services that are provided by nature that we don't worry about and we don't even cost. Well, availability of water is one such thing, but in this context, the pollination services. Now, if there is an environmental degradation, I think the whole entomological system may change and may affect pollination. And now the question is, so even otherwise, there is a cost, environment and health. So are there methods of costing? Number one, predicting what is going to happen in next couple of decades. What is the net damage in economic terms? And what are the ways of factoring that in terms of the costing structure to make sure that the environmental conservation actually gets the resources that are needed? Because otherwise it's kind of, when there is a lot of popular pressure, you put that, and I'm not too sure that whether this money is utilized in a balanced way. It is utilized for a good cause, I know, but whether it is utilized in a balanced way. So now you may say this is too much of a problem and it cannot be done within the within the domain of just ARI, which I'm also very conscious. And I'm also very conscious that I don't even know, of course, I'm not a person in the field, but even if you want to define a roadmap for solution to this problem, I don't know whether we have the ability to define that roadmap. But I know that this problem is very important. And I also know that ARI can make a big contribution, if not by itself, but certainly in collaboration with a number of institutions. Many of them are in Pune, but even those who are outside, within the country, outside, wherever. So can we now think of a network program where there are a large number of projects with their own very specific, clearly spelled out objective and definition, but they are all linked to this addressing this larger problem, and then approach the governmental bodies to kind of take that mission. It could become a mega project for us to handle. And I can reel out many examples, and I'm not going to kind of take time. But for example, bioenergy is a forte for you. I know today the biomass, particularly after the kind of development of 2G and other related technologies, the agricultural residue, and that is surplus. Mention was made about rice, but surplus agricultural residue, which after meeting all other requirements, there is so much of residue that if you convert that into ethanol or any other fuel hydrocarbon, you can actually replace the gasoline requirements. And at least the import of hydrocarbon equivalent of gasoline consumption in the country, that import can be saved. Now, that is the level of this. And it is for this reason that Government of India is giving so much of emphasis. That is, that statutory requirement of ethanol blending is only a small factor, but in reality, this can make a huge difference to maybe around 25 to 30% reduction in the foreign exchange expenditure for import of fuel. That is the potential of this. And I know, because I'm connected with these activities, today, in spite of giving so much of thrust to bioethanol production, the cost of bioethanol is commercially not viable. It is all, initiatives have been made because government is creating subsidy and government is pressurizing PSUs to kind of take the setting up of these plants. So a dozen plants are being set up in the country, but it is for us, the researchers, to make sure that this technology becomes commercially viable. So like that, I can reel out in everything. I just want to give one more example and then I'll move to another track. And that is, I was very impressed about the cultivar, the fortified wheat variety that has been developed at ARI, MACS 4028. Now, as was mentioned, this of course is very important for the nutritional kind of making India nutritionally self-sufficient. And even within that, there are various micronutrients. You're talking about iron and zinc. But iron, I know for sure, and actually I'm connected with a project which is running at Derwent, the Valavalkar Medical College and Hospital, where they are looking at the anemia-related issues among adolescent girls and the issue of the babies born to them later on and their health status. And there is reason to believe that this is one single factor which can make the whole generation, kind of, in terms of instead of becoming a contributing generation, it could become a whole generation which will become a drag on the society. And that's the level of importance of nutrition. And so such studies require long, something like 15, 20, 25 years surveys. And to some extent, on a limited scale, they have been launched at Derwent. And nutrition supplementation is an item as a part of this study. So I'm going to refer this to also the people there at Derwent, and they will contact you. But I think use of this wheat could also become a part of this larger mega study. And so you can see, you could have a collaborative program from agriculture on one side to addressing for example, the problem of non-communicable diseases in the young population, and which has, it is an intergenerational issue, and it is, to my mind, it is connected with the future of India. And so can we define some projects at that level on an inter-institutional basis and get it done? So I don't want to take too much of time, but I think every activity that you're all engaged in, to my mind, looks extremely important. And I also know that ARI has recently signed an MOU with University of Pune, and this academic network called setting up collaborative activities and all. I think such inter-institutional formal arrangements are extremely important. And so I think the future lies in group research, not only in terms of supporting fundamental research or basic research, because you could have these big problems in basic research which will require group activity, multiple projects led by multiple group leaders and large number of scientists. But also in case of translation, where you have to collaborate with people of different kinds, different expertise, different ways of working. For example, the project that you're dealing with, energy, will be one such area. So both, whether it's connected with translational research or connected with addressing larger fundamental issues, I think group research becomes extremely important. And so let us see whether we can expand this group research to from within the institution or small groups across the institution to much larger groups which can address big and scale problem. And that, I think, according to me, that's the pathway to scale up and also go to higher level of excellence. The last point that I wish to make is one of this very key feature of ARI IC is the kind of, of course, you have a peer community also within the institution, but you also have a large peer community consisting of people who are retired from the institution but are still connected with the institution, and still larger peer group because you are located in Pune. Now, I think, I'm sure there are lots of interactions taking place between this group and the institution. But I think this we must recognize that this has a great mentoring role in terms of, creating ideas, or if you have an idea, creating a roadmap, how to go about it, who are the people to contact, how to get into a network, because these people have great ability to connect. And so we probably, and of course, also in terms of evaluation of performance of programs, evaluation of performance of individuals. So when we sort of get into actual thing, I think we should try to understand what is the present situation in this context, and how can we go forward, because these are the ways to move towards the higher level of excellence. So I know I'm taking too much of your time, and I think looks like more than half an hour past the allotted time. But I am so happy that we have a great team, both in the governing body as well as in the larger peer community of the institute and the community within the institute itself. And I think together uh, we can certainly uh, walk on a maybe an uncharted territory, but that's what research is about, but towards great objectives and great heights. So I want to wish all of you good luck and great success in scaling these new heights. Thank you very much.
Thank you so much, sir, for your very encouraging words and your guidance. And I'm sure we are all re-energized, rekindled with new fire to walk the steps that you have shown us. And before we close for the day, I would now invite Dr. S.K. Singh to officially propose a vote of thanks. Dr. S.K. Singh, please.
Thank you, madam. And good afternoon to everyone. Respected viewers, I am Dr. S.K. Singh is profusely elated to propose a vote of thanks. Today, being the Founders Day, is an occasion to commemorate our research achievement, and we are indebted to the visionary who founded the MSCS ARI way back in 1946. Every year, we celebrate this occasion on a larger scale, but this year, due to the COVID-19 pandemic, we had to restrict this program. But again, this year is very special for us. ARI has entered into the 75th year, and the Department of Science and Technology has entered into its 50th year. Through the initiative taken by the Department of Science and Technology, its science popularization arms, that is Vigyan Prasar, made the film on Nagarkar Research Institute, which you have already seen. So DST Vigyan Prasar needs a special mention for releasing the film today on the occasion of our Founders Day. One more notable development at our institute is the formation of new governing body of MSCS led by respected Dr. Anil Kakodkar sir. I thank you, Dr. Kakodkar sir, for your valuable guidance. Sir, under your able guidance, ARI will further progress and excel in all the thematic area of research. As you suggested that we have already planned to various activity which will be undertaken round the year as a mark of celebration of our 75th year celebration. On behalf of ARI and on my own behalf, I thank you for your sparing valuable time to preside over this function. I thank you, Professor N.R. Kalmarkar sir, the Vice President of MSCS, for sparing your valuable time and expressing your thoughts. I thank Dr. Vidya Gupta Madam, Secretary MSCS, and Dr. S.M. Ghaskarbi sir, the Treasurer MSCS, for your gracious presence, sharing valuable thoughts and extending valuable support to ARI. I also thank Dr. P.K. Dhakefalkar, Director ARI, for being a driving force for extending all kind of support to organizing this program. And at the end, I thank all the viewers, scientists, staff, students, unit in charges of ARI for their direct and indirect support in organizing this online program. And I must congratulate our scientist colleague from botany for bringing our very informative books. Once again, congratulations. And before final conclusion, I would once request all the viewer to watch this movie on on your own device, because this active link will be active, because I felt that there were some disturbances and time lags when this movie was showing during this program. So you will really enjoy the movie. So once again, thank you. Thank you very much for one and all.
Thank you. We can leave, right?
Yes. Yes, sir.
Thank you.